Now, the second part of what we need to discuss regarding blood has to do with kind of the more application and real-world type uses and workings with the blood. And that has to do with blood typing. Now, you have to keep in mind we have 4 basic types of blood: A, B, AB, and O. And these are actually determined by genetics. We have 2 copies of a blood typing gene, one from mom and one from dad. And these genes exhibit dominance. In other words, type A is dominant over O, which is recessive, and type B is also dominant over O, which is recessive. But that AB occurs because we also have a codominance situation. In other words, we have both genes, both A and B, are dominant, and they are both expressed. So we see the actual individual traits of both. So there's no blending. If you think back to your sophomore level genetics, this would be the difference between like having something with red spots and white spots, which is codominance, versus blending, which is incomplete. So red and white would make pink. So these individually express with no blending. So the blood type is given to the name of the protein that is the antigen, and that exists on the surface of our red blood cells. So we have a type A protein and we have a type B protein. Type O has no proteins, as we will see. In addition, with our blood and the proteins that are present, we also have to look at the plasma. The plasma contains antibodies that will actually attack the protein that is not present on the surface of the red blood cells. This causes what we call agglutination, which is basically clumping. This is clumping that is different than clotting. We don't have the clotting cascade getting initiated with this agglutination. Simply, it is the red blood cells actually getting sticky and sticking together. So, for example, type A blood has the A antigen but B antibodies. Type B has B antigen and A antibodies. Type AB blood has both antigens but no antibodies. And type O blood has no antigens but both types of antibodies. And you can actually see this represented kind of in picture fashion here in this diagram. So type A, as you can see, has the kind of V-shaped antigen on its surface, which according to the key represents protein A. And then the dark circles that are around it would represent the antibody Bs that exist in their plasma. Now, if you look at type B, it has a different shaped antigen, the type B protein present, and a different shaped antibody, antibody A, that's around it in the plasma. You can also see here that the antibody here with A would actually fit in kind of lock and key mechanism into the antigen and the protein on A. So that's what I mean by it will attack the protein A, whereas the antibody B won't. Those don't fit into each other. Type AB has both types of proteins on its surface with no antibodies, and type O has no proteins with both antibodies. So let's kind of take a first look here at these terms antigen and antibody, because we will use them here, but we'll also use them in the lymphatic system when we talk about immunity. So an antigen is basically a protein that acts as an identifier. Sometimes I like to call these the name tags. They say, hi, I belong to, whoever, Susie. And all of Susie's cells are going to have identifiers on them that actually show that those cells belong as part of her body. Now, cells that don't belong as part of her body will not have that the right protein, the right name tag. So they actually stimulate an immune response, kind of like the bouncer trying to kick them out. Antibodies are those protectors. Okay, they are the body's defense against a foreign antigen. So what they do is they recognize ones that should not be there, and they actually try to stop them from continuing to survive in our bodies. Antibodies are what we call antigen specific. In other words, you have to make a specific shaped antibody to counteract a specific foreign antigen. As you saw in that diagram, they fit together. The more antibodies that your body produces, the more easily you are able to fight off an infection or disease, because you've got a bigger army, basically, to attack. Now, making sure you have the right blood combinations is important in the medical community, simply because we deal with things that are called transfusions. And transfusions are basically the input of blood from a source other than yourself, usually, for purposes, to combat either some sort of disease or condition, usually that is temporary. Sometimes it's done to basically save your life. So this compatibility of the blood is important during a transfusion. If this compatibility is not correct, it can actually cause what we call a transfusion reaction. So transfusion reaction is caused by receiving the wrong type of blood. And what types of blood that you are able to receive depends on your blood type. All right, so let's first look at what happens in a transfusion reaction. Let's say that we actually have received the wrong blood type as part of a transfusion. What exactly is going to happen? And then we'll talk about what types of blood are compatible with each other. The first thing that happens is we get agglutination, that clumping of the blood cells. The antibodies that are present in you as the receiver's blood will attack those foreign red blood cells, and it causes them to get sticky and clump together. These little clumps clog our small blood vessels all over the body. So it starts with the capillaries, and then it'll move into the arterioles and then the venules as well. Now, this clotting actually isn't the problem, because what it does is it actually keeps those foreign cells from moving, and it makes them really easy targets for those white blood cells. As part of the attack by the white blood cells, these red blood cells, these foreign ones, are lysed. Basically, lysed means to cut or rupture, and when you basically pop open a red blood cell, it spills its guts, which is all hemoglobin. So now you have this dead cell and this free hemoglobin that gets released into the bloodstream. Free hemoglobin is bad. You do not want free hemoglobin running around in your bloodstream. What this does is it actually travels in the bloodstream to the kidneys, and it actually blocks the capillaries of the kidneys. This leads to kidney failure, because the capillary of the kidney is actually where the filtering takes place. So you prevent the kidneys from actually filtering the blood. So the kidneys shut down, your blood is not able to be filtered, and you actually get this buildup of waste material in your blood. It can lead to death if you can't get your kidneys refunctioning, or you don't do some other artificial type of filtering like dialysis. Now, some other symptoms that can occur with this, and it's not a guarantee. If you just get a little bit of the wrong blood type, so there's a lot of factors that are involved. How much blood did you receive of the wrong type? How soon did they catch it? That sort of thing. This is the worst case scenario. Some other symptoms that can occur are kind of like if you would get sick. You can get fever, chills, nausea, and vomiting. None of which are pleasant, but obviously it's a lot better than dying. Okay, so let's tie together the idea here of the blood type and the transfusions that take place. So, the first thing, if you look at this table that's in your notes, it has some other terms that are there that we typically will not use. And it's just mostly for consistency's sake, from me to you. We're not going to use the term agglutinogens. We're just going to use the term antigens. To me, it's actually easier because you guys know, well, you will know what an antigen is between this unit and the lymphatic system, and I think you've got enough vocabulary to worry about. The other one we're not going to use is the agglutinins. We're just going to use the term antibody. So, with blood type A, as you can see on the chart here, the type of antigen that is present, that protein that is present, is the A antigen. So for type B, we have the B antigen, and for type AB, we have both A and B antigens. Type O, as we saw in the diagram, has no antigens or no proteins present on the surface. Now let's take a look at the plasma. Type A blood has the anti-B antibodies present in the plasma, and type B blood has the anti-A antibodies. Type AB can have none of the antibodies, because if it has either one with both protein types present there, it would actually attack its own blood, and that's bad. Type O, since we have no proteins present, has both anti-A and anti-B antibodies present. Now, we talked about this idea of transfusions, so let's take a look at the different combinations of donations and receiving that we can have. Now, when you donate or receive blood, you can always donate or receive from your own type. So for instance, type A can donate to itself, type B can donate to its own type, type AB can donate to its own type, and type O can donate to its own type. We know that. Now, let's go back up to A. If I donate type A to type B has this anti-A antibodies, which are going to attack the proteins on my type A. So I can't donate to there. If I take type A and I donate it to type B, there are no antibodies present to attack it. So I can donate to type AB. If I were to take type A and put it in with type O blood, the anti-A antibodies in type O would attack that blood. So we don't want those attacks to happen. So type A can only donate to A and AB. Type B blood, it cannot donate to type A because of the anti-B antibodies. If it goes into the type AB, because again it has no antibodies, it will not get attacked. If I put the type B blood into the O, the anti-B antibodies in the plasma of O blood would attack it. So I can't donate to there either. So just like type A, the only ones that type B can donate to are its own type and type AB. Type AB, as it's listed here on the table, can only donate to type AB blood. If I put it into type A, it has the anti-B antibodies, which will attack, and type B has the anti-A antibodies, which will attack. Type O has both, so none of these three can I put type AB blood in without having a transfusion reaction. Now, type O blood has no proteins, so it can donate to itself, but it can also donate to all of the other blood types because there is nothing that can get attacked. Since there is no antigen on its surface, the antibodies that are present aren't going to attack those antigens. All right, in terms of receiving blood, blood can be received just like it can be donated from its own blood type. Now, we have to look at this kind of in the opposite way. If type B blood goes up and gets donated to type A, okay, the anti-B antibodies are going to attack. So we can't can't receive blood from type B. Same thing with type AB. These anti-B antibodies will attack the B antigen on the type B blood or type AB blood, so I can't receive from AB. But type O will not get attacked, so it can be received by a person with type A. Same parallel situation, okay? With B, it can only receive from itself or type O. Type AB, because it has no antibodies in its plasma, can actually receive from A, there's nothing there to attack it. It can receive from B, there's nothing there to attack it. And it can receive from O, because there's nothing there to attack it and there's nothing to attack. Type O, if I put type A in, the anti-A will attack. If I put in type B, the type B will attack. And with AB, both of them will attack. So O can only receive from O. Now, you would think that the low amount of O in terms of receiving would be a problem. Fortunately for people with type O blood, the majority of the human population actually has type O blood. So that is typically not an issue for people with type O blood. O is called the universal donor, and that is because it has no antigens to get attacked on the surface of the red blood cells. Type AB is referred to as the universal acceptor since its plasma contains no antibodies, so it will never attack any other type of blood that is given to it. Now, a lot of times I get questions from students who say, well, I'm AB positive. What does that positive mean? We haven't talked about that yet. Well, that refers to what we call Rh factor. And Rh factor, Rh stands for rhesus, for the rhesus monkey. This is actually a trait that we share with our closest primate relative. And this is where that comes from. Rh factor is what gives that positive or negative notation to our blood types. Now, this is a different antigen than with your blood. Okay, so it's separate. If your blood cells have the Rh antigen on their surface, it's considered to be positive. If there is no Rh antigen there, then you're negative. So positive, it's there; negative, it's not. Now, this becomes important for both blood transfusions, and we'll kind of talk about how this works, and during pregnancy or childbirth. Now, with a blood transfusion, if someone is Rh negative, and it has to be that you are Rh negative. If you're Rh positive, you will never get a transfusion reaction. But if a person is Rh negative and they get Rh positive blood, the first time, nothing happens. And the reason why is because their body does not have any antibody in the blood. So, in other words, okay, if this is their red blood cell, okay, and we look at the Rh negative, there's no protein present there. Whereas with the Rh positive, we have that protein on the surface. Now, if we were talking about the 4 blood types, the Rh negative would already have antibodies running around in the plasma against that protein. But that's not how this works. What happens is my blood does not have any antibodies against this Rh protein. But what happens is when I'm exposed to this, my white blood cells that produce antibodies learn how to make the antibodies. So now, over a little bit of time, my blood actually acquires those antibodies, and they stay present in my blood. So what happens then is I get that buildup of antibodies, and so if I receive an incorrect Rh match the second time, I get a transfusion reaction. So it goes through the agglutination, and it can lead to kidney failure. Basically, with Rh factor, you get one, I kind of call it the get out of jail free card. You get one opportunity to screw it up. If you screw it up more than once, you're going to have a problem. With blood type, in terms of the blood type compatibility, you don't get that free pass. Now, in childbirth, this also becomes an issue. And again, it only is an issue if mom is Rh negative and whatever first baby, that comes along that is Rh positive. This is what starts the process. The first time, nothing will happen. What happens during childbirth is when the placenta detaches from the uterine wall, you do get some breaking of capillaries on both the baby side and mom's side. And mom's and baby's blood mixes a little bit. On mom's side, that's what triggers the buildup of the antibodies. Okay, so if she gets pregnant with a second Rh positive child, her body, the antibodies actually go across the placenta and start to destroy the infant's red blood cells. They start lysing them. This causes a condition known as hemolytic disease of the newborn. Basically, the red blood cells of the infant can get destroyed in such a way that it not only can cause things like anemia, which we discussed earlier, but it can also create a condition called hypoxia, where the brain is actually denied oxygen. This can lead to brain damage or potentially even death, just depending on the aggressiveness of those antibodies. Now, this used to be a very scary condition, but what we have is a RhoGAM shot. Basically, this is one of the reasons why babies' blood types are determined right after birth. If mom is Rh negative and it's determined that baby is Rh positive, she will actually receive a shot. It's called RhoGAM. And what that does is it shuts off mom's system from producing those anti-Rh antibodies. So every time she has an Rh positive baby, she's going to need to get one of these shots. Just because it suppresses her immune system from carrying out its normal natural process. Otherwise, with any subsequent pregnancies that she has, she runs the risk of having her baby either become a miscarriage or stillborn or have some serious problems when it is born.